So the Blood-Wise Clarity study was, is one of the first trials to combine two novel targeted agents together in relapsed or refractory chronic lymphocytic leukemia, or CLL. So we treated 50 patients with relapsed CLL with a combination of ibrutinib and venetoclax, so targeting the two main pathways that are abnormal in CLL. Our main aim of the trial was to eradicate detectable disease, both in the blood and marrow, and that was at 12 months. Whilst the key secondary endpoints were safety, to make sure there's no unexpected toxicities, and eradication of the disease at 6 and 24 months. And the plan was always to use that data to modify our next phase 3 trials. So we recruited 50 patients, and we reported last year the safety, that this was a safe thing to do. We had no increased risk of tumor lysis syndrome or any other particular side effects. They were just what we expected with the two drugs alone. And we're now reporting the first early response data, showing that we're managing to eradicate detectable disease in about a third of patients at the 6 months of combination. And we're seeing responses in almost all the patients, well, in all of the patients, with usually the bone marrow being clear in the majority from a morphological point of view. One of the exciting things, I think, in CLL is a rapid change, both in the understanding of the disease. So we have a lot of molecular techniques that have really been applied to CLL, and we're really starting to understand the biology of the disease. And also, in sort of parallel with that, the increase in the numbers of therapies. So in the first until about 10 years ago, we only had 2 or 3 different types of treatment, but now we have many more that are developing, and that's given us a great opportunity to move the treatment forward and to actually even think about curative approaches with combinations, but also the challenge of trying to get the trials recruiting rapidly enough and working rapidly enough to get the results out before the standard of care changes. So what we've managed to do in the UK is to use the TAP program, the phase 1, phase 2 program, to test the safety and the efficacy in patients who have failed treatment before, and then to rapidly take those therapies into the frontline setting. So using an adaptive trial design to allow us to add new treatments to the phase 3 program. And I discussed that in the meeting today. I think what we're seeing in CLL is not only the acceleration of the trials, but also the translational research. So the fact that we're biobanking samples, we're doing analysis and looking for resistance mechanisms. So I think we're learning from the trials, and therefore adding and using novel treatments as we see that the trials are patients who aren't responding as well. And so I think over the next few years we're going to see an acceleration of that, and our aim is to eradicate detectable disease in as many patients as we can, because that's means that we can safely stop the treatment and have patients off treatment for prolonged periods of time and potentially cure patients. The PSH this year has been very exciting. It's been exciting because of the sessions I've been to, the interaction between the NHS and how the NHS is working to facilitate novel agents going to trials. So we're in a very privileged position, I think, in the UK, that we have the opportunity to put most of our patients into trials, and we're using that to attract sort of resource and drugs into the country. So I think we had talks from NHS England, from the NIHR, showing that we're gelling these things together, and also from the molecular side of things with Genomics England. So I think the whole picture of interaction and integration of the science and the trials and therefore the treatments is really exciting.